Welcome, and thanks for joining this presentation on pathway and network analysis in bioinformatics. Over the next few minutes, we'll explore how computational methods help us interpret biological data, identify key molecular players, and guide research in areas like drug discovery and precision medicine, all while keeping explanations practical and focused on real analytical approaches. This slide outlines the journey ahead. We'll start with a brief introduction to bioinformatics, then move into pathway analysis methods, network analysis techniques, strategies for integrating multi-omics data, illustrative case studies, and finish with future trends and challenges in the field. To begin, let's set the stage. Bioinformatics sits at the intersection of biology and data science, turning raw biological measurements into meaningful insights. In this section, I'll explain the core ideas and why computational analysis is essential for making sense of modern high-throughput experiments and translating findings into biological understanding. In simple terms, bioinformatics develops tools to analyze biological data and solve problems in genomics, proteomics, and more. As shown in the illustration, it combines computer science, statistics, and biology to manage large data sets and reveal patterns that drive discoveries in drug development, agriculture, and environmental science. At the core of pathway and network analysis are three ideas. Pathways are ordered biochemical routes. Networks are complex interconnected systems, and the components, genes, proteins, metabolites, interact in diverse ways. Understanding these concepts helps us model biological systems and interpret how multiple pathways coordinate cellular behavior. Why run pathway and network analyses? They help pinpoint key regulators, genes or proteins that control processes, provide a holistic view of complex biological systems, and enable predictions about system behavior under different conditions, which in turn can guide experiments and accelerate discovery. Now we shift into pathway analysis methods and tools. In the next few slides, I'll summarize common approaches like overrepresentation analysis, functional class scoring, and topology-aware methods, explaining when each is useful and what trade-offs to expect when applying them to genomic or transcriptomic data. Overrepresentation analysis, or ORA, identifies pathways that contain more genes from a given list than expected by chance. Tools like David, GOSEC, and Cluster Profiler make ORA user-friendly and database-driven. But remember, ORA focuses on counts and can miss magnitude of change, sometimes leading to false positives. Functional class scoring evaluates whether genes in a pathway show coordinated expression changes, with GSEA being the most popular example. This approach considers the magnitude and direction of gene expression, making it more sensitive than ORA, but it requires genome-wide expression data and can be more computationally demanding. Topology-based pathway methods incorporate the structure and connectivity of pathways, not just membership. Techniques such as SPIA combine enrichment with pathway wiring to detect more biologically relevant impacts, offering deeper insights at the cost of needing detailed pathway maps and greater computational complexity. Turning to network analysis, we'll cover three major network types used in biology: protein-protein interaction networks, gene regulatory networks, and metabolic networks, highlighting how each is constructed, the typical data sources, and the kinds of biological questions they help answer. Protein-protein interaction networks are built from experimental or curated interaction databases and reveal how proteins physically or functionally connect. Resources like String, Intact, and BioGrid power these maps, which are invaluable for finding drug targets and understanding protein function within disease mechanisms. Gene regulatory networks map regulatory relationships between transcription factors and target genes. Common inference methods include correlation analysis, mutual information, and Bayesian networks. GRNs are useful for predicting gene expression programs, identifying key transcriptional regulators, and studying developmental or disease-related regulatory changes. Metabolic networks represent enzymatic reactions and metabolites, connecting pathways of cellular metabolism. Databases such as KAG, Metacyc, and Recon3D provide comprehensive reaction data, and these networks help identify metabolic biomarkers, guide engineering strategies, and illuminate disease-related metabolic shifts. Next, we'll discuss integrating multi-omics data, a crucial step for gaining a comprehensive view of biological systems. Combining genomics, transcriptomics, proteomics, and metabolomics helps bridge layers of regulation and offers richer biological interpretation than any single omics type alone. Multi-omics integration faces three main challenges: heterogeneous data formats and measurement scales that complicate integration, computational scalability issues when handling large datasets, and the complexity of interpreting results, which demands cross-disciplinary expertise and careful biological context. Common methods for multi-omics integration start with rigorous data normalization and transformation to make datasets comparable, followed by statistical approaches like correlation, regression, or PCA to find relationships, and, finally, network-based integrative methods that combine different network types for a holistic systems view. Practical use cases of multi-omics integration include cancer research, where combining data can reveal tumor heterogeneity and treatment targets; drug discovery, where integrated profiles help predict efficacy or resistance; and personalized medicine, where individual multi-omics signatures guide tailored therapies. Now we'll move into case studies that show these methods in action and how they translate into research and clinical insights. In cancer research, pathway analysis can reveal dysregulated signaling routes in tumors compared to normal tissue. For example, KEEG pathway analysis in lung cancer often highlights PI3K ACT and MAPK signaling as frequently altered, which provides leads for targeted therapy development and hypothesis-driven drug discovery. In neurodegenerative disease research, network analysis of protein interactions has been used to investigate Alzheimer's disease, revealing interactions between APP and tau proteins and other partners. Mapping these networks helps identify potential therapeutic nodes that could slow or alter disease progression. Multi-omics integration is increasingly used in precision medicine to tailor treatments to individual patients. By combining genomics, transcriptomics, and proteomics, clinicians can build a more complete profile of a patient's disease, informing treatment choices while acknowledging the need for standardized pipelines and responsible data handling. Looking ahead, the field faces exciting opportunities and hurdles as technologies and analytical methods evolve. In this section, I'll highlight emerging trends in algorithms, data practices, and ethical considerations that will shape how pathway and network analyses contribute to future biology. Advances in algorithms and tools are accelerating research. Machine learning can uncover complex patterns in biological data. Cloud computing provides scalable resources for large analyses. And improving user interfaces makes these methods more accessible to experimental researchers, broadening the impact of computational approaches. Data availability and standardization are key to progress, promoting responsible data sharing while safeguarding privacy. Adopting standardized formats to improve interoperability between tools and databases, and encouraging community-driven efforts, like consortiums, to facilitate reproducible integrative analyses. Ethical considerations must guide our work, protecting patient privacy through robust security measures, addressing bias in biological datasets to ensure fair outcomes, and responsibly using analysis results by carefully considering the potential societal and clinical implications of our findings. Thank you for watching this overview of pathway and network analysis in bioinformatics. I hope these summaries help you understand the main methods, their applications, and the future directions of the field, and that you feel ready to explore these approaches in your own research.